come back to that. We have all these different input factors, and then it's connected through mathematical equations to a hidden layer, and then it comes out to the output function. So you start out with this data. These are real world data that you enter about your trees, and in our system we're looking at some 450 trees. And then these come in as your input, and then it looks at the output, the fruit yield. Okay, was the answer correct or not? Okay, if it's not correct, or if it's off, then it comes back and it reweights all these different linkages until it figures out what are the most important variables. And the output then tells you, it gives you a sensitivity analysis, tells you which one are controlling it, and then you can set, at that point, each of these at desired levels, and then the computer is now recognizing these patterns and coming back and giving you an actual predicted output or an association. Typically it takes about 5,000 steps, but this is, or you could have a few hundred steps depending on how simple the data are. But this is the same way that our brain works. We smell smoke, we see light, we feel heat, and go, oh, fire, we make a decision. Some things are weighted more than others. And this is also figuring out the audience. You can see, okay, male, female, how old, interest, tired. Our brains do these things, pattern recognition. And this is the basic idea behind the neural network model. Now in our study, we're working with some 12 locations that span the entire zone of the avocado production. Depending on the year, different numbers of trees in production, but about 450 trees in the entire study. Typically in any one year, only about half our trees are yielding. This is interesting. Carol Lovett finds the same thing in her data sets. So alternate bearing and just other things going on. Some trees are always low producers, others are good producers consistently. And we're working across a variety of sites that have different management techniques, slopes, soil types, rootstocks. We have 5 central locations where we have more than one rootstock, and at those we have soil water data. And then we have the other satellite locations where we're collecting the leaf nutrient analysis and the yields on individual trees, so we can tie a leaf nutrient to a particular yield. Now the idea behind this again is that if you were to control any one single variable and all the others were held constant, then you can come up with these types of relationships. For example, you could look at the pH effects. Low is good, high is bad, and there's a curvilinear relationship. You could look at salinity effects. You could look at nitrogen effects. And all these will have different effects on yield. You put them all together and you get something that looks like this. Now, how would you ever sort that out? You couldn't through a traditional statistical analysis. And that's exactly what we find when we look at avocado. You predict yield versus leaf chloride, and you get a scatter diagram that looks like this. And you say, well, there is no relationship. I could never predict chloride was causing a decrease in yield based on that data alone. Have zero relationship. These exact same data, you run them through the neural network model, and we're using, in this case, a program called Synapse. We put in all these different variables on the soil type, pH, organic matter, water quality. We look at the type of rootstock, the leaf nutrient contents, chloride levels. We can then go into different things on the output, looking at yields of primary interest. We've looked some at root mass. We can calculate water use efficiency. We can look at alternate bearing, fruit quality, lots of different variables, all based on these as input to predict the output. And doing that, this is what we find. That same diagram you just saw that was a scatter diagram converts into this. Predicted fruit yield, kilograms per tree, for the different rootstocks, Mexican, Duke, Savandusa, and Toro Canyon. And here we see that the Dusa and the Toro Canyon are the top producers under low chloride conditions. That Dusa falls off pretty quickly at leaf chloride levels of 0.6. It would predict no yields. Toro Canyon continues to perform out here with some yields down at 0.98. So now we can get an actual numerical quantity of the effect of chloride on avocado yields. Now this is based on this model. We turned all those little slide bars. We had adjustments for each of those tools where we set it to the mean for the entire study. And across the entire, all the trees, 450 trees we looked at, the mean was 2.4% nitrogen. Phosphorus is 0.18, potassium 1.2, and calcium 1.5, et cetera. And that's what we get. What would happen if we went back in and adjusted those slide bars to maximize the yield, and then tried to see the effects of chloride again. It's the exact same plot, but in this case, we have brought the nitrogen down to 1.7%, brought the phosphorus up to 0.26%, and those are the main two switches that greatly improve the yield in the system. And here we see for these same rootstocks, even the Mexican rootstock is still producing where Toro Canyon was previously out here. Before we had complete failure above 0.8 except for Toro Canyon. But now we see by optimizing the tree's nutrition, we're able to obtain yields. And that gives us some guidelines. Are there some specific chloride recommendations that we can make, or fertilizer recommendations for trees subjected to chloride? And I think this can also be a very useful tool for looking at reoptimizing, redetermining these critical nutrient levels in the plant tissues. This was largely developed in recommendations on citrus. And so I think using these types of approaches, we can now begin to look at the relationships between these elements, the nutrient ratios, and the effects overall on yield, optimal levels. This gets back to your nitrogen question. And here we have a nitrogen interaction with chloride. And in the model, what we're looking at here is 0.1% leaf chloride on this level going up to 0.8 over here on this end. And you can see that under low leaf nitrogen, we end up dropping off in this profile. Now, as you go up in leaf nitrogen, the best yields you can see actually curve up into here, where we're getting some 600 pounds per tree. And we could even tolerate 0.4% chloride levels if we keep the leaf nitrogen somewhere between 2 to 2.4. And then after 2.4, it starts to drop off. But as you go up to 3.2, 3.4%, as long as the chloride levels are low, you'll keep some of your yields. But if the chloride levels go up to 0.8, and then you also increase the nitrogen up here to 2.8, you're in big trouble. You've lost a lot of yield. So you have a nice vegetative growth. You probably still have a lot of leaf burn because of that high chloride level, but you've really ruined your yields. So now we can start to look at these interrelationships in great detail. And again, they're associations. It's just data that has been associated, not necessarily cause and effect, but at the very least, it guides our experiments and things that we can do to try and confirm those relationships. This is for calcium and phosphorus. This one's also very interesting, again showing fruit yield here for the leaf calcium levels and the effects of chloride. And what we can see is that as you increase the leaf chloride, or leaf calcium levels up here to 2.5%, we almost completely eliminate the toxic effects of calcium, or of chloride on the system. Over here, we look at the relationship between nitrogen and phosphorus, and bringing the phosphorus levels up also are very important, and they can offset some of these detrimental effects of having high nitrogen in the plant. So these are the things that are coming out of real-world data. If you went out and looked at these trees, these are actually representing trees that are out there that have these levels of nutrients in them, and those would be their yield performances. Now, going back into the literature to try and see, well, is there a basis for these things that the model is finding? You can go back into the 90s, the 80s, and the 70s when a lot of traditional plant nutrition work was done with tanks and so on, different very detailed experiments looking at individual nutrients and their effects under precise conditions. And here we find results from one review article. This is a 1990 review. It says, nutrient additions have been more successful, such as correction of NA-induced calcium deficiency. Here, Champagnol reviews 17 publications find phosphorus added to saline soils increases crop growth in 34 of 37 plant studies. Most cases, salinity decreases the concentration of P in the plant. P additions may be required with salinity. Great. We also see here, what about calcium? As the salt concentration in the root zone increases, plant requirement for calcium increases, going back to 1970s. Another report by Moss in 1993 at the Salinity Lab, maintaining adequate calcium's important factor in controlling specific ion toxicities, sodium and chloride injury. And in citrus, calcium was found to be effective at reducing transport of chloride from the roots to the leaves. So what we're seeing is that the model is actually predicting what took decades of research to discover in horticultural plants. And that just by feeding in these data from the field, it can recognize these same patterns. So I think it's very exciting to see things that we know being confirmed and then being pointed in new directions that might help to guide our better management.